Hey guys, it's Miss Arneson. I'm here to walk you through cell specialization for Science 142. What the cell? We're going to talk about cell diversity real quick. Diversity means a lot of different types. And so this is what we've been talking about so far. This is a generalized cell. But most cells in the body actually don't look like this at all. They have really specific shapes and they have specific jobs. Here, we just make this model of a cell, this generalization of a cell, so it's easier for us to understand what all these different parts are, kind of what they look like and what they do. But if we really start zooming into the human body or any organism that's multicellular, then we're going to have a lot more complicated structures going on. And so this, for example, is a neuron, and it does not look like this at all. Here is a blood cell. Blood cells, red blood cells don't even have DNA in them. They don't even have a nucleus. When you do forensic testing, you actually test the other cells in it because they do have DNA in them. So even though we learned that the nucleus is like a big deal and it does everything, basically directs all of everything, some cells don't even have a nucleus. So not very many. So cells can be all different types. These are layers of skin. And so, of course, these are different shapes than the blood cells, different shapes than the neurons. And we're going to look at how does this happen? Like, how do these become this way? You're supposed to be taking notes during this lesson, but you don't have to take notes on this page. Just pay close attention. In reality, life is pretty complicated. This is a picture of a cell, and this is a zoomed in picture of basically the same type of cell. And you can tell they're complicated. These are black and white images, so this is like what we would take a picture of under a microscope, a really good microscope. And it shows us some details about things that we can't see with our naked eye. But it's kind of hard to understand what's going on here. Like if you were going to ask me, and I'm a science teacher who's been studying a lot of things before, can I identify any organelles in this picture? I would be like, this looks like a nucleus. Like that's all I could get. So we have to make models to help us understand things better. Here's reality. This is a zoomed in picture. If you ask me to identify organelles in this one, I might say, I don't know, this kind of seems like it might be a vacuole to me because it's like, it looks like an empty space and vacuoles are meant for like holding things and storing things. This I know is a mitochondria because that's how they look. I've seen a lot of pictures. But it doesn't really help me understand what's going on in here. So instead what we do is we have models. They represent reality, but they're not as complicated. They're simplified. And of course, they're not always going to show exactly what happens in reality because they're going to be generalized. So here is a false color image. So this isn't reality anymore because we've taken these colors and we've assigned them to certain parts of the cell so that we can see the differences between the different materials here much more easily. And then this is a drawn model. It looks computer made to me, but it has this whole part right here called rod outer segment. There's also rod inner segment. So I didn't learn about those on the generalized cell. Endoplasmic reticulum, mitochondria, Golgi apparatus, nucleus. I did learn about those. So those are things that I can recognize and like basically already know what their job is. These other parts, these specialized parts, I'm not sure what they mean. So let's talk about, take a second to talk about why we do models. Oh, here's I wanted to zoom in on that eye for you and show you how many different layers of different types of tissue there are just inside this really thin layer around most of the back of your eye. It's pretty nuts. Okay, so this is a model that you saw a lot at the beginning of COVID. Basically, they were saying if we do nothing and we don't have people social distance, this is going to be how many people get COVID. And all of these people that are above this red line, there's not going to be enough healthcare services for them. So their chances of dying are going to go way, way up. So instead, what we need to do is have interventions, and people will still get COVID, but they'll get it at a lower rate. So they won't all have it at the same time. And that means that our healthcare system can take care of them and we don't have to have just people dying because there's not enough doctors, there's not enough ventilators, there's not enough oxygen. This was a computer model that generated a graph for us to look at. We use models basically all the time in science, and there's a bunch of reasons why. The purpose of them is to help us comprehend what science we're talking about. So if you're talking about something really complicated like the parts of a photoreceptor rod, which was that last cell you looked at, then it's easier for you to understand the different parts of it if you break it down and you draw a picture of it and you label the parts of the picture. There's also really difficult things that it makes us more able to comprehend, like the size of the universe. You have to make that with a model that shows things in relative sizes. Otherwise, you just can't even think about it because it's just too big and too old of a thing for your brain to process. We can use it to make predictions and test things. That's really big. So, depending on the type of model that you're using, like a physical model or a two-dimensional model, which would be like a drawing, with those ones, you're showing it to represent reality. Like if you draw a cell model, for example, that would be two-dimensional, and you're just representing what reality looks like to simplify it and help you comprehend it. So that would be going with the comprehension part. But there are other things that are really complicated in the universe that we need to be able to test some other way than actually going out and doing it. For example, you cannot go out and figure out how the dinosaurs died by just doing an experiment in your backyard. Why? Because you can't have dinosaurs in your backyard and you can't be a giant meteor that crashes into asteroid really, that crashes into Earth and like kills all the dinosaurs, right? You can't recreate that situation. So instead you have to use something that represents that. And in that case, you could use a sandbox to kind of show what happens when you drop a marble, the asteroid, into the sand, the Earth. It puts a whole bunch of sand everywhere. Anyways, we also can't test certain things because it would be unethical. Like on human beings, you can't just run tests on human beings. So we need models to help us figure out what would happen in human beings. The different types of models there are mental or visualization. This is crazy to me, but old textbooks used to have basically no pictures. And I mean, I can't even imagine that because textbooks now have a billion pictures. But what you used to have to do if you were reading a textbook is make a mental model of what you were reading. You'd have to see it, visualize it in your mind. And I wonder if we had less pictures, if we would be better at it, because I feel like a lot of people have gotten worse at visualization. But like if you have a daydream, you're technically visualizing. So you can do it. You just have to practice more to get really good at doing it for science things. And then you can have computer models, which is like the weather. Of course, computer models aren't always right. Like, I'm sure there's been days when it's told you it's going to rain, doesn't rain. And you were like, dang, the computer model of the weather was wrong. But that's because reality is more complicated than any model can make. So that's why models aren't, if you can study something in person, you always want to do it. But sometimes you can't. And then physical models we discussed, like the cell model. Of course, physical models, and then there's also visual models where you draw them. Okay, let's talk about what happens when a cell's going from being like a generalized cell to a cell that has a special job and looks very different than the generalized cell. So there's going to be three main steps of this. If I were you, I would write like, I would make a box, like one box, two box, three box across your page, and then draw arrows between them. But maybe actually draw the box after you write the words because sometimes you don't draw the boxes big enough. And the first box is going to say stem cells or progenitor cells. Okay, so you start off this process with stem cells or progenitor cells. These are a little bit different, but for us in this class, we don't have to really like focus on the differences between them. But stem cells are the type of cells that can turn into a bunch of different types of cells. Progenitor cells still can turn into a bunch of other types of cells, just not as many as a stem cell, but they're both doing the same thing. So you start off with this cell and it doesn't have a job yet. So it could become a skin cell, it could become a blood cell. Okay, you have this cell that could be turned into anything. What happens is that that cell, and this is in our second box, so make sure you write down progenitor cells, which is spelled right here, Progenitor, and stem cells in this first box, and what they are. They are cells that can be turned into multiple specialized cells. So they're kind of general. In this middle part, what's going to happen is that genes will get turned off. Genes will get turned off. So even though your muscle cell and your neurons, the cells in your brain, they look very, very, very different. they still have the same DNA in them. You could still take one of these theoretically and clone a whole nother Miss Arneson from one of my muscle cells. Thankfully, we don't do that. Although it would be nice for me to have a housewife that could like cook and clean, and then I come home from work and it's already done, but it was me, you know? I don't. That's a fun sci-fi. Think about that. Okay, so these cells have different jobs. The reason they are shaped differently, the reason that they function differently and do different jobs is because the genes that don't need to be expressed, the genes that don't need to code for proteins, get turned off. So that's the second step. Genes get turned off. The process for this, which we'll talk about it later, but we're just going to name it right now. The name of the process that turns off these genes that aren't needed is called methylation. It's METHYLATION, methylation. So these cells are going to get methylated, and depending on what the body needs, different types of cells will be made. Isn't that crazy? So, one of my questions for this class is, why can't I smell with my butthole cells? The reason is because the smelling proteins are not coded for and not made in my butt. And I'm really grateful for that because I don't like the smell of poop. And I have a really good nose. Like, I have a really good sense of smell. But anyways, what are these 3 steps? You start off with a progenitor or stem cell. Genes are going to get methylated. If they're methylated, they will not code for a protein. And then you get cell specialization on this last part. The cell will do something special, it'll do a specific job, and it will be made a specific way, depending on what proteins get coded for. Okay, so this right here, stem cells, is something I want to talk about because it's been in the news a lot over my lifetime. Maybe you've heard about it, but probably COVID news has superseded that in importance. You start off with peeps, okay? They have sex, they get their sperm with their egg. Those are their gametes. This little N represents that it only has half of the DNA, right? So it only has 23 chromosomes. Then when you get an egg with 23 and a sperm with 23 and you put them together, you get a zygote, and then you actually have the amount of DNA, 46 chromosomes, that you need to make a person. So after that, what happens? Okay, because that's where we got to basically in class so far. Well, this is going to tell us about the next step, cell specialization, cell differentiation. Those are 2 words for the same thing. Mitosis is going to happen, and these cells are going to start splitting. So this is just a microscopic image of a fertilized egg that has started to divide, and it has a bunch of cells. So this is by day 7, day 8. So you can see it's starting to grow more different types of things. Right now, it looks like there's this one little dark spot, and then there's a bunch of these like really clear looking cells, but you start getting differences. Like this looks like it has little layers now. I don't know if this picture shows up very good for you on the video. I hope so. But by the time you get down to like day 18, day 15 is the one where I see the most detail. There's a lot of different stuff going on in here. There's like layers inside of this that are all doing different things. Those are specialized cells. So they're still undergoing mitosis, they're still copying themselves, but now genes are starting to get turned off so that these cells can be turned into any part of the body. This right here is a model just to help you guys see what's going on in these pictures a little better because it's got color-coded cells. So like day 7 and 8, these are the 3 different types of cells that are in there. But if you get all the way to day 16, now we're looking at 1, 2, 3, 4, 5, 6, 7 different types of cells, and that's because they're specializing. So it takes 9 months, of course, to turn that into a baby. All right, guys, talk to you later. Have a good day.